We're going to pick up here with electrophysiology. One of the characteristics of our cardiac muscle is autorhythmicity. That means it sets its own rhythm. It doesn't need any interaction from our nervous system. It is an unconscious activity for us. Those cardiac muscle cells contract when they are excited by electrical excitation, and this would be your action potentials. And unlike smooth muscle cells and skeletal muscle cells, these cardiac cells don't require any stimulation to have these action potentials. They handle it by themselves. It's coordinated by a small population, about 1% of our cells in our heart, that are called pacemaker cells. And these cells will rhythmically and spontaneously create these action potentials, and then that spreads to the other type of cardiac muscle cells, the other 99%, which is your contractile cells. The cardiac muscle cells, as we learned back in the tissues unit in chapter 4 of 201, they have striations. They have the alternating light and dark bands, and that's because of the way the contractile proteins are arranged, and that's the characteristic they share with skeletal muscle cells. These cells are branched, and they have one nucleus, and they are much shorter and wider than our skeletal muscle fibers are. They have a lot of myoglobin that carries oxygen, and they have very high energy demand, so they're going to have about half of their cytoplasm is filled with mitochondria. They also have those intercalated discs to join the cardiac muscle cells together. And it also joins the pacemaker cells to the contractile cells. And these intercalated discs have the desmosomes that hold the cells together, and also the gap junctions so that ions can pass from one cell to the other, so that all of the cells are communicating with each other. If you look at them, you can see here at the top how it's branched. And this is the intercalated disc that you see there that has the desmosomes and to hold them together, and then the gap junctions so that different ions can pass from one cell to the other. You see there's a lot of mitochondria in these cells, and that's because they need a lot of ATP, because they're always contracting. Like the skeletal muscle fibers, they are excitable, and they have these gated ion channels in their sarcolemma. And the way these ion channels open and close, that's responsible for all the action potentials in the pacemaker cells and the contractile cells. They have a specialized cardiac conduction system, which is a group of interconnected pacemaker cells, and those pacemaker cells will rhythmically and spontaneously depolarize to cause the action potentials. The types of gated channels, you've heard these before. There's the voltage gated sodium ion channels. Whenever there's a voltage change across the membrane or a gradient, they're going to that's what maintains it, and these sodium ion channels will open, and the sodium can cross across the membrane. And you find these in all of the contractile cells, not the pacemaker cells. The calcium ion channels, they also are voltage gated. However, they are time gated. In other words, they close at the end of a certain time regardless of the voltage. And all of the cardiac muscle cells, the pacemaker and the contractile cells, have potassium ion channels that are ligand gated or voltage gated.